Hello, everyone. Good afternoon, and welcome to another Science Division Live. This one is called From Specimen to Species, How to Determine Hard to Distinguish Species. And we are back today with Tiffany and Andie up in our Genetics of Taste lab. They are so excited to present some stuff about Colorado's chipmunks today. So if you've ever spent the day in Colorado's mountains, which I hope you have, you might have seen a chipmunk scurrying along the trail. But did you know that there's 5 distinct species of those little tiny little chipmunks, and they're hard to tell apart? So we are going to go under and behind the skin of the chipmunks and find out how DNA sequencing can help scientists crack the code of those 5 separate species. So go ahead and take it away, Andie and Tiffany, up in the Genetics of Taste lab.
Hello, everyone. My name is Andie, and I'm the Zoology Preparator over in the Zoology Prep Lab here at the Denver Museum of Nature and Science.
And I'm Tiffany Nuessle, and I'm the Research Manager in the Genetics Lab here on the second floor. This is where we actually are today. You can see our lovely sign.
Like Elsa said, we're going to go through how our 2 labs are collaborating to distinguish really hard to determine species. In this particular display, we're going to show you how we distinguish between chipmunks. Chipmunks are really hard to identify just by looking at their physical features. So we need the help of genetic analysis in order to help us figure out which species is which. So this presentation was done for Girls in Science back in March, and you might have been there, you may not have, but we're going to show you a little bit of what we did for that event. Let's get started.
Okay, I'm going to go in and share the screen.
Oh, and if you have any questions, make sure to ask those in the chat. We'll be doing a Q&A afterwards at the very end. Once we get our mystery species done. So we're going to start off by going over the biological species concept. The biological species concept focuses on physical features of animals. So here we have 2 different types of chipmunks. Both of these occur in Colorado, and both look very, very similar. So you have Tamias quadrivittatus, also known as the Colorado chipmunk, and Tamias umbrinus, also known as the Uinta chipmunk. So these 2 chipmunks, they look very similar, and they also overlap in their range. So they live in the same areas. So using the biological species concept, we'll use the physical features that we see on these skins to try to identify both. So there's a few different ways that you can a few different characteristics that we look at when we're trying to identify chipmunks. One is the color. So you'll notice that the Tamias umbrinus, the Uinta chipmunk, is a little more orange on the sides than the Colorado chipmunk. You'll notice that the tail tip colors on the Colorado chipmunk is also a little more tan colored than the Uinta chipmunk, which is more white. And then you can also take a look at the color of the feet. The Colorado chipmunk is a little more drab versus the Uinta chipmunk, which is more orange. And then the last coloration difference that you might see are the stripes. So a lot of times you can tell chipmunks apart based on how bold their striping is. So in this case, we notice that the Uinta chipmunk, the middle stripe goes all the way up to the top of the forehead, and it's pretty strong. The Colorado chipmunk has a weaker stripe. So those are just some very basic differences that we'd see. On top of that, you can also take a look at measurements, and there's a lot of field guides that will go over ranges, measurements, and any other distinguishing characteristics. So for this exercise, we're taking a look at these 2 different chipmunks, and let's say that we go out into the field and we see another chipmunk and we want to identify it. So we have this little mystery. So you can use the coloration, you can use some measurements to try to identify this one. In this case, you'll notice that its feet are a little drab. Its dorsal stripe, so that middle stripe is a little more like the Colorado chipmunk, but it's very orange on its sides, so it looks a little bit like the winged chipmunk. So I want you all that are watching to take a look at this mystery species, and try to identify it yourself. Is it a Colorado chipmunk or is it a winged chipmunk? Using both coloration and all the little details that I just talked about. So I'll give you all a few seconds to take a look at it, and just to make sure that you have time to really get your answers in. All right. So once you've figured out which one you think it is, we'll go on to the next step. We'll actually go on to the taking tissues and trying to identify through genetics. So we can get up and do that. So I will.
Okay, we're going to take just 1 second, and we are going to be switching to the other speaker so that we can follow Andie along on, I can follow along with the phone while she does a demonstration. So just one moment.
Sounds good. Everybody watching, go ahead and throw your questions about these chipmunks or about DNA sequencing there in the comments section, and we'll try to answer some of those questions as soon as we are finished with this demonstration. All right. Andie, that looks great. Go ahead. Yeah, that sounds good.
One second. There it goes. That's it. Yeah. All right. So I'll have Tiffany, my camera person, and perfect. So you can just tilt it down so you can see, they can all see the. Great. Wonderful. All right. So when we're actually starting off the genetics process, the first thing we need to do is we need to take tissue. So we have our little chipmunk stuffies here, and we have a little vial ready for our tissues. There's certain tissues that we take in the lab. We take heart, liver, muscle, and kidney, and each one is great for very specific reasons. So kidney and liver are really great for toxicology studies. And then heart and muscle have a lot of mitochondrial DNA, and we can trace the mitochondrial line with those. And then for just regular old DNA, you can pretty much use any of those, but we typically use muscle for the genetics lab. So what would happen is you search around, you try to identify some tissues. In this case, we have a little bit of lung. You'd put it in a tube, just like that. Secure that with a top. And then you'd have little labels. So I'll just show you what those look like. So part of the label has a specimen number, and we'll just assign that a random number of our choosing today. Let's say my initials. So we'll do A, M, C, and then the date for today. So today's June 11th, so let's say 0611. And then the species, which is right in there, will get filled in later on when we actually go through the whole DNA extraction and replication and barcoding process. And then we'll circle in the type of tissue that we have. So in this case, we have a lung. So in the end, the tube is nice and labeled, and it would go up into the genetics lab so that Tiffany and the people that work in this lab can do the extraction, replication, and sequencing of the DNA.
Okay, I'm going to hand this back to Andie and move on over. So you can see our lab in the background. This is the genetics of taste lab. Go ahead and mute on yours. Okay. Are we back? Okay, perfect. So once we get the DNA or the tissues from Andie's lab, our goal is to separate the DNA to make a lot of copies of it and then to send it off for sequencing. Now, before we get into how we do that, I just want to go over a little bit about DNA. So here at the museum, we like to compare DNA to a cookbook for your body. So this is your little body's cookbook. And I'm going to go to share screen to show you a couple of the inside pieces because I just don't think that this would give you the quality you need. I think the computer will work a little bit better for that. So when we're talking about DNA, DNA is like the cookbook for your body. And a gene then is like a recipe. And just like a recipe has ingredients, your genes have something called a codon. And those, the way that we write out ingredients in a cookbook is we use the letters of the alphabet. And in the same way, we use something called nucleotides or letters of the DNA alphabet to write out our codons. Now, what's really amazing about DNA is you actually only have 4 nucleotides. And a nucleotide is a fancy word for chemical. And they are A's, T's, G's, and C's. You can see them right here. And when we combine 3 of them together, we make a codon. So doing the math, that means there's only 64 possible codons or 64 ingredients that can make up your gene. And putting them in certain orders changes so much about you. But here's what's so fascinating. Changing one codon may or may not make a big difference to a recipe. So for example, if I'm cooking and I'm making these chocolate chip cookies and I run out of butter, but I have margarine on hand, it's a substitution, but it doesn't really change much. Most of you probably wouldn't notice, some of you probably will, but I'm the type that won't. It is a little bit different, but it's still going to be a basic chocolate chip cookie and delicious. But some substitutions, some ingredient changes make a huge difference. For example, if instead of chocolate chips, I put in raisins, one ingredient changed, but most of you are going to feel like it's an entirely different cookie. And so when we get to speciation, sometimes we're looking for some of these really big changes. And then some of the littler ones might be within one species of chipmunk. It'd be more like the butter and the margarine changes. They might be kind of similar. So we are going to now switch over to what we actually do once we get the tissue. So we're going to do one more flip and go from the screen share back to the computer because we like to mix it up here. Okay, so stop share. Are we good to go?
It looks great.
Perfect. So the first thing we have to do with the DNA is we need to separate it from the rest of the tissue that we had. And I'm going to show you a little bit about what we do. So here at the museum, we have an experiment where we actually do this with wheat germ. But Alyssa has put in the comments section here a way to do it at home with strawberries. So using all household ingredients. All you need is baking soda, meat tenderizer, a little bit of liquid soap, some water, and then some isopropyl alcohol, which you may or may not be able to find right now. But what we will do is we will mix in some of these ingredients and we break open the cell and that releases the DNA. But at first, when we do this, the DNA is released, but if you can look in here, it just kind of looks like a little pile of goop. And that is because everything is in here. There's all the cell debris, there's the proteins and all sorts of other stuff. We need to pull out the DNA. And that's where we add the isopropyl alcohol. So DNA isn't soluble in alcohol, which means it's going to stay. So you can see all of that floating up top. It's really kind of cool, like a little jellyfish or something right now. That is DNA. Oops, let's see if I can get it in focus. That is the DNA from the wheat germ. And if you unravel it, it's about 6 feet long per cell. It's so amazing. That's at least human. I don't actually know for wheat germ how long it is, but we'll see. We'll go with 6 feet. So once we have it separated, we have to make a lot of copies. And the way that we do this is so cool. Okay, so your DNA starts off in this shape. It's like a double helix. So if we unravel it, it kind of looks like a ladder. And what you're going to notice in looking at it is that the green ones, here those are guanines, are always connected to a blue one. And the red ones are always connected to a yellow. Now, the green might be on top or the blue might be on top, but they're always together. And this is what happens in your DNA. A C and a G always bind, and an A and a T always bind. This becomes so important because it means that what we can do is actually split apart this helix into a single strand, and we automatically know what the other side should be. So we're going to run through that process. And this is called polymerase chain reaction, or PCR. So we're going to come right over here now. So we have your DNA. I'm going to tilt this a little bit. Right, there we go. And what we are going to do is unzip it. And when we do that, you can see we only have one side of the ladder, but we automatically know what the other side is. If I see a blue here, a cytosine, we know it's a guanine on the other side. If I see a yellow thymine, we know it's a red adenine. And this is really important when it comes to the PCR. So we're going to come right over here. I'm going to show you a little bit. Okay, perfect. So in order to do PCR, can you tilt it up? Just thank you. In order to do PCR, we have to add a couple of things, and I'll go over those in a second. We have to add the DNA. And this is what we would have just extracted from the wheat germ. We have to add little primers, which are short segments of DNA unique to the area we're interested in. And then we add something called master mix. And that has both a bunch of free nucleotides, like right here, just like floating around, kind of like a little alphabet soup. And then something called a polymerase, which I'll get to in just a second. So PCR is a series of heating and cooling. And what we do is when we heat up the DNA, we break the hydrogen bond. And all of a sudden, we have two single strands of DNA. And when we start to cool it, it starts to rebind. But here's where the primers are so cool. Because they're so much littler, they can move so much faster. And they'll actually bind to the DNA before this connection can happen again. So we put in two primers, one for this strand and one for this strand. So for example, I have that primer attached here, and I have another primer. I'm going to move this so you can see, attached right there. And then we have a polymerase. And the best analogy I can think of here is this is like a zipper. So if you have gotten a zipper here, you can go along and you're going to pull in the free nucleotides. So we're going to do that for just a second. This zips along and it just starts finishing out what this strand would look like. So going through here, and it's going to continue along. And we get to the point where it's doing this on this side as well. And then we have a complete strand of DNA, but we don't actually have one, we have two. So say I've completed these, I don't want to take all the time to do it. Then we have the two strands. Then we heat up the DNA again. And we break them apart again. And we just keep going through this process. Every single time doubles the amount of DNA that's in there. And so we do this 30 to 40 times. So we make gobs of DNA. And that's so important because we're going to send it off to somebody and they're going to be looking at it and they're going to be determining for our species, the one we don't know, what letter they have in certain areas, what nucleotide. So I'm going to show you quickly what PCR actually looks like when we send it off. When I talk about making gobs and gobs of DNA, we're talking about in tiny, tiny, tiny little areas. That's it. Now, just as a caveat, DNA is not green. DNA is clear. However, the master mix that we add that had all the free nucleotides, the primers and the polymerase, that is green in this case. And so that way we know if we've done it. So then we send it off for sequencing. In this case, we send it to a place in California. And it's just cost effective for us because they can specialize in that. And then we can get it back and we can do the analysis. So now I'm going to go back. We're going to go back to screen sharing. Okay. So I am going to get my little sequence and we're going to switch back to screen sharing one more time and we're going to go over that.
That's great. We do have some questions, so get ready, you guys. It's going to be great. Everybody is really curious to know which species we have now.
Okay, well, once we have our sequence, I'm going to give you a second just to take a quick look at it, see if you can memorize it. No problem, just 4 letters over and over and over, different patterns. So we're going to go back to the screen share, and I'm going to jump ahead just a few spots. And these are the sequences that you might see for the quadrivitatus and the Tamias umbrinus. And we're looking for the differences. As you can see, these are actually fairly similar sequences, but there are 3 variations between them. And that's what we want to do a comparison of. So I'm going to show you one more time in here. I'll zoom in closely so you can see it. Take a second and see if you can figure out which species it is. Okay, so now we're going to move on. And who knows what our mystery chipmunk was? It's the Tamias quadrivitatus, or known as the Colorado chipmunk. And I'm going to turn it back over to Andie at this point, who knows a whole lot more about these chipmunks than I do. And she's going to share with you a couple facts about this particular one. Stop sharing.
Yeah. So yes, our final chipmunk was Tamias quadrivitatus. So these chipmunks are really interesting. They're found mostly in Colorado, hence Colorado chipmunk. And they tend to overlap ranges with the Tamias uinta, or the Tamias umbrinus, or the uinta chipmunk. So they can be really hard to distinguish, but luckily with the genetic barcoding, we are able to tell the difference. So the Colorado chipmunk, the way that they breed and the way that they find mates is actually really interesting. It turns out that the females get to choose, and the females are actually attracted to the fastest males. So the males will display by running around everywhere, and the females will choose the fastest one to breed with. And sometimes that ends up being a chipmunk of a different species. So at times you'll see the Tamias quadrivitatus and the Tamias umbrinus that overlap in ranges will actually breed together and make hybrids. So that's pretty cool, and that's another reason why we do this genetic analysis. Identifying where those hybrids are and studying the behavior can be really benefited by genetics. So yeah, and that's our presentation. Does anyone have questions for us?
We do have questions from the chat. Andie, maybe this one is for you. You say heart tissue is great for obtaining mitochondrial DNA. Someone was listening very closely. Does the proportion of mitochondrial DNA actually differ between tissues within an individual's body, or is it just the degree of expression in your DNA?
Yeah, that's a great question. So depending on the cells, you can have different types of organelles or little baby organs that are found inside the cells. And heart muscle does have more mitochondrial DNA than regular muscle or any other organ. So it's really great for tracing that mitochondrial line. So other tissues that would have like specific types of organelles or large amounts of a specific organelle are like livers, kidneys. They have what are called lysosomes, or like dumpster organelles, because they're used in filtering blood and detoxifying your blood. So each specialized cell can have different types of quantities of organelles within each cell. It's a great question.
Oh, it was a great answer too. Thank you. All right, Tiffany, Rachel asks, how are the tissues stored once you get them in the lab?
So we actually keep them down in the vertebrate prep lab until we are ready for them. So we store them once they are prepared in a -80 freezer. And at that point, though DNA will still degrade, it is slowed down so much in that process that we can store the DNA for such a long time. There are other ways that it can be stored. Sometimes there's liquid nitrogen or other things used. But for right now, we use a -80 to prepare it. Once we have extracted the DNA, so as I said before, when we separate it out, the DNA is in alcohol because that helps us we end up dissolving it back into a water for long-term storage. And then at that point, we keep it in a -20 freezer until we're ready to send it off for sequencing or do any more work. Great question.
Wow. All right. One more question each. Andie, what about the 13-lined ground squirrel? Where does this fall into place with your chipmunk?
So the 13-lined ground squirrel is also a rodent. So they're both in the rodent family. They're somewhat related, but not quite. So they're both ground squirrels. Few people know that chipmunks are actually ground squirrels. But we haven't done any work on 13-line ground squirrels here. I'm sure people have done genetic studies on them in other places, but they're, for those of you that don't know what 13-line ground squirrels are, they're probably the prettiest ground squirrel that we have in Colorado. And they're really awesome hibernators that their limbs can go below freezing and they don't get freezer burn or frostbite. So they're pretty awesome little guys. You should definitely check them out and Google them when you're done watching this live.
Awesome. Thank you. All right, Tiffany, we had one viewer who said they would definitely want to purchase this cookbook for your body because if you compare everything in my body to cookies, I would also buy that. But one of our, Karen wants to know, what's the current broad definition of a species or of speciation? Because some species are defined more softly. So can you give us some background, some information about speciation?
I can give you limited background on species work. So just to put this in perspective, I've been working with the animal specimen for really only the last year, 7 or 8 months. If you see up above here, this says Genetics of Taste Lab. Prior to this, we've been working on human taste and genes related to there, so we weren't doing any species difference. So I have been getting a crash course on some of the animal knowledge. I can share a little bit, but my guess is actually Andie might know it better than I do. Maybe not. We'll see. Okay. So the thing is, there are still a lot of there's a lot of uncertainty out of how to define different species. So one of the most common perspectives is that it's when two animals can mate and produce a viable offspring, and then that that offspring could continue the line. That's one of the most common ones I've heard, but I know that there are many theories. I was reading a paper by one of our curators, Dr. Garth Spellman, who is our curator of ornithology, and he was writing a paper that talked through a few different theories dealing with migratory patterns, and I didn't know there were so many different theories, actually. So contact Dr. Garth Spellman to ask about speciation, or Dr. John Demboski. I will happily throw them under the bus for this. It is an intense but really cool conversation because there are a lot of ways, and this is part of what shows that science is still evolving. We don't have a great, well, that I know of, let me preface, necessarily have a really amazing definition yet.
That's great. Thank you both so much. We are out of time for today, but we hope you all join us again tomorrow for another Science Division Live. Go ahead and keep those comments coming in the comments section. If you have more questions, just let us know. Otherwise, we'll see you all tomorrow, and thank you for joining us. See you later, everybody.
Take care.